20th, right? Yes. What happens 1 week from today? Okay, exam 2, October 27, Friday, from 12:45 to hopefully about 2:15. And it'll be up in McCormick, was it second floor? Weekly. Pardon me, weekly, second floor. What was the number? 030, 031, 032. Okay. And we also know now that the final exam is going to be go over in the ISC building, and you'll get more notice of that later on. The reason I'm excited is because it's a lot nicer space. Okay, so that'll come up, and I'll be sending you out a message as to what it covers and that sort of stuff. Okay, the other thing is that we're in the season where you guys who are still continuing, anybody graduating in December? Congratulations. Have you got your plans for what happens afterwards? Talk to career planning and placement, talk to your advisor. Make some plans if you haven't made them. But next spring, you guys got to register over the next month or so. In a conversation out in the hall with a person who shall remain nameless, it was brought to my attention that there's a fallacy out there. And I'll dispel the fallacy. Okay? Juniors and seniors in good standing aren't limited to 300 and 400 level biology courses. Besides looking at other departments like environmental science, or physics, or math, or chemistry, you can also look at 400 and 600 level courses. Those are grad courses. Okay? A lot of them have interesting topics. There are a lot of them that are run with seminars, and a few have labs, not all of them. Okay? So if you're in this boat and you haven't found something that really fits in with what else you're taking, you're looking for something to fill in or finish up, look at those courses as well. You may find something. The one thing you have to do as an undergrad is to get in contact with the instructor and get a permission number to register. Okay? I was also asked what I'm teaching next spring. I am not teaching endocrinology in the spring. I am teaching mammalian toxicology, which I've taught some before, but which is going more and more toward the use of bioinformatics. It's a seminar and lecture. There's no lab with it. And the number is Biology 666. It's the study of poison, so why not give it the devil's number? So I just wanted to announce that. And if anybody's interested, just get in touch with me. Okay.
So is it like pharmacology, or?
I think I stated earlier on in the term that endocrinology, pharmacology, and toxicology are sister disciplines. They use much the same information because they're all based on assays and biological responses. Endocrinology deals with endogenously produced chemicals that largely maintain homeostasis in the body. Pharmaceuticals, it deals with the response to to man-made and naturally occurring substances that may improve the body's performance or the body's response in conditions of deficiency or disease.
And this is all the detrimental stuff.
And toxicology deals with the largely negative effects of synthetic and naturally occurring materials on the body, and how we measure them, how we get it, or what we get into with respect to regulation, and how you cope with 10,000 new chemicals a year that need to be tested before they can be used in anything that touches humans, animals, the environment. That's a pretty large task, and there's a large backlog of chemicals that need to be tested. At any rate, so if anybody else had those questions, hopefully those provide some answers. Today, I want to take us on a journey into steroid biology. Steroids were among the chemical signals that were isolated largely between about 1920 and 1950. And they're lipidic or lipid-like compounds. They're largely hydrophobic. And they may be considered what are called secondary metabolites. That is, they're small, they're produced by enzymatic processes from substrates that are available either in the diet or are made in the body. But they're very different from what you saw with respect to protein hormone formation. Protein hormone formation, transcription, translation, post-translational processing to get these molecules into circulation and for use as intercellular chemical communication messages. So, and there's a lot of stuff about steroids in the news, so we'll do some exploration of this system. Your recording as well. There's at least 2 or 3 other people recording, so you have to get in touch with them. Okay. Right. Now, if you notice this slide,
No, I see. So sad.
that's obviously an old slide. Okay, an old picture anyway. Steroid functions. You may have run across this in your reading, but I'll reiterate it. There are 3 or 4 main functionalities that steroids perform in a physiologic system. And steroids are found in virtually any vertebrate system. In fact, they're found in some invertebrates as well. Glucocorticoids and sex steroids support growth and development, especially in the embryo, gonads, brain, and pubertal animal. Okay. The glucocorticoids help maintain glucose homeostasis and modulate stress responses. Does that sound like something you've heard before? Yeah. Okay. We're going to reinforce that. Mineralocorticoids help maintain sodium balance and blood pressure. Okay, we haven't gotten into that. We'll get into it a little bit more as we go through the term. Sex steroids regulate and maintain gametogenesis and reproductive function. In that list, are any of those steroids potentially life-threatening if you remove them? Which ones? Homeostasis and stress response. So the glucocorticoids. What about the mineralocorticoids? Okay. So you have to have a balance in sodium and blood pressure. What about sex steroids? Can you get along without sex steroids?
All or one?
All.
Yes.
She says yes. Who says yes? Who says no? Who says yes? You don't have too many backers. Who says no? Okay. Who says, I don't know and I don't care? Okay. Well, think about it. There are individuals who are born without gonads. Okay? There's agonadal individuals. Do you think those folks live any shorter period of time than others? Don't know? Okay.
They're alive, that's the question.
Okay. Does anybody here know what a eunuch is?
Yes.
What is it?
In Italy, they really liked the sound of young boys' voices, so they castrated them before puberty so that they would maintain their falsetto.
Those were castrati. Those are castrati. Those are prepubertal
That's what I mean.
castrations. Not necessarily. A eunuch is a castrate male. Okay? So, if they are castrated before puberty, there's an interesting correlation with that castration. They live longer than other males.
Whoa.
Okay?
Wow.
Like sad life, right?
Pardon? We can talk about that later. Okay. So yeah, sex steroids are not necessary to support life. The other steroids do. Okay. They cover a broad spectrum of physiological functions, and over and underproduction causes potential problems. If you've got production of steroids in general, you can get delayed or precocious growth or development. Overproduction of glucocorticoids particularly can lead to diabetes. Overproduction generates an abnormal immune response. If you get an underproduction or problem with mineralocorticoid metabolism, you're going to get high blood pressure. Female infertility, endometriosis, that is inappropriate growth of the lining of the uterus. Cystic ovaries, ovaries that have large but afunctional follicles, ovarian failure, hirsutism. What's hirsutism? Anybody? Pardon? Hair in the face. It's inappropriate growth of hair. Okay. Early pregnancy loss, menopausal symptoms. What's osteoporosis? What about bone? Decalcification in bones, so thinning of the bones. Okay. Lactational failure. All of those are associated with sex steroid production, inadequate sex steroid production. Male infertility, spermatogenic insufficiency, erectile dysfunction.
Professor,
Yes?
the osteoporosis, is that specific to female steroid insufficiencies, or?
You can get this out of a combination of other things, but it's most associated with estrogen insufficiency. Okay. All of these conditions with respect to the male. What's prostatic hyperplasia? I know there's some of you guys who work in the cancer labs. What? It's enlargement due to what? What's hyperplasia? You're going to need to know that next week. Yeah, it's an overproduction. It's a rapid division of cells. Often you end up with smaller cells than normal. Okay, but it's an inappropriate growth. It's usually the lining of the prostate in this case. And prostatic hyperplasia, prostate cancer is going to occur in something over 80% of males who live beyond year 80. Okay. It is very, very common. It's driven not just by testosterone, but by a hormone that's derived from testosterone called dihydrotestosterone. It's much more potent. But the androgens drive that process. Part of the reason that eunuchs live so long, they don't have to worry about that. Reproductive cancers of the breast, ovary, cervix, endometrium, testis, and prostate. are all sex steroid related. Yeah.
Androgen or term meaning sex hormone?
No, we'll get to that. Okay. Gonadal anatomy. You're going to see this again, but I'll give you the tour a little bit to begin with. I want to turn this light off so you can see the slide better. This is would be testis from a rat. What you're seeing here, the big bulb is the testis proper. It's encased in a sheath-like covering called the tunica albuginea. This slice of tissue, including this bulb down here and including a bulb on the other side, the other end, is the epididymis, where a lot of the maturation, the later maturation of sperm occurs. And then up here, in this, tucked into this fat pad, are a lot of blood vessels and the vas deferens, which leads up to the upper male reproductive tract. Of course, spermatogenesis and steroid production, androgen production, the major processes going on in that gonadal tissue. In this slide, oh, I can sit down for this. In this slide, what you're seeing are a slice through a testis, and it's showing you the seminiferous tubules, the places where sperm are actually being produced. It's also showing you scattered cells in between, in those spaces. Leydig cells, the cells that actually produce testosterone, are found out here, often around the blood vessels. So there's Leydig cells here, there's some in here, 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 here. The Sertoli cells are the nurse cells, and those make up a lot of the cell mass that you see here around the interior periphery of the Sertoli or the seminiferous tubules. These tubules have a basement membrane around them. If you take them out of the testis without fixation, they'll come out as a loose mass. It looks like spaghetti, quite literally. I haven't tried cooking it, but you notice that the inside pattern in the lumen of these tubules differs in these various tubule segments. And we'll talk about that more when we talk about spermatogenesis. But at this point, just to let you know that these are actually various stages of the spermatogenic process that are being pictured here. So this one would be an early stage, and the late stage is where you get the tails showing up as the primary pattern in the middle of the tubules. These little black dots around the outside are the spermatogonial stem cells. So these are the stem cells that give rise to the sperm, which are in the middle. And the Sertoli cells, a lot of this tissue out here. Okay. On the female side, this is a female, I believe this is in a cat. What you're looking at is a structure. Here's one that is not, where you don't have much follicular growth going on. In this one, you've got one follicle that's grown much more rapidly than the rest of the follicles in this organ. And this would be a pre-ovulatory follicle, so the next time this animal ovulated, the egg would be coming from that follicle. This structure up here is essentially this structure after follicular rupture and under transformation to the corpus luteum. Slice through a side of one of these things, you'll see that an awful lot of follicle growth occurs just under the cortex, or the outer layer of the ovary. There's a lot of blood vessels coming and nerves coming in from what's called the hilus down here. But a lot of the growth of germ cells, and remember there's one germ cell in each one of these follicles, That's the ovum. And then there are granulosa cells. And then on the outside, you see a few of them that look like they have a dark green. That's thecal tissue just to the outside of the growing follicle. Okay. These making estrogen and progesterone primarily. Yes?
What determines where the follicle forms?
In large part, what determines is how many other growing follicles are around it and how well you got how much vascularization you have in the area.
So there's room for it and something to sustain it?
Yeah, whether there's room for it and whether there's competition for growth factors and nutrients by growing follicles around it.
So that organ right there is an ovary?
This organ, okay, each one of these is an ovary, so one's the right and the one's the other left, and then this is an ovary down here. It's cut off over here, which you can see it would come around. The shape isn't too dissimilar from a kidney. The size of the ovaries, in a primate, the size of the ovary is about the size of the end of your thumb. And the follicles, the lead follicles, and the size of a full-blown corpus luteum is about the size of your thumbnail. Somewhere around 3/4 of an inch to an inch across.
So that corpus luteum, once it fully goes down, then there will be no more follicle.
Well, the corpus luteum is post-follicle.
Right. So there will be no more, like no eggs descending from that.
No.
section.
This structure, its job is to make progesterone, to support implantation of fertilized egg. Okay. The adrenal, which we talked about a little bit before, this is a typical picture of what you'd see if you open an animal and we're looking for the adrenal gland. Here's the top hole of the kidney, and then the adrenal gland is a real fatty tissue that sits up here. Okay. They're asymmetric, so the shape is a little different on the right and the left. They have a core, and again, that's neurally derived. And then this outer portion is where all the steroids are being made. And this is just another picture. This one happens to be of a sheep adrenal. And you can see a lot of vascularity in this area, less so in the middle, and very little actually on the outside. This is a picture from a rhesus monkey, a section through. Here's the medulla. And then you've got the zona reticularis, where you're getting a lot of androgens from, weak androgens. And then fasciculata, glomerulosa, where the fasciculata is producing a lot of the glucocorticoids, and the glomerulosa is where the mineralocorticoids are being made. And this is just another picture of the various layers and blown up. And you see they differ in terms of their structure. You've seen these axes before. You're going to see them again. Got that? Okay. And so, they differ obviously between the male and the female in terms of the production, in terms of their controls and which elements are involved. And then the ACTH down here. These are the controls for steroid production in mammals. Okay. Now, before I get to this, I want to take a little bit of a side journey. If I can do this. Yep. Come on. And what I've got here is a slide that talks about a process called receptor-mediated endocytosis. You'll get And you'll get the reason why I put it up here now in about 5 minutes. Okay. There are many molecules physiologically that carry precursors or nutrients or that are protein hormones. And in most of those instances, those materials bind to receptors on the surface of the cell, and those receptors, either prior to binding or sometimes after, collect in areas called coated pits on the surface of the cell membrane. And those coated pits have a protein, a structural protein underneath called clathrin. And those are areas that will eventually invaginate into the cell and be pinched off as coated vesicles. Those are endocytotic vesicles. There are vesicles coming from the outside of the cell to the inside of the cell. And once they enter the cell, a number of things happen. These are the earliest stages of the endosomal lysosomal network, organellar network in cells. They will lose their clathrin coat and become early endosomes. And as they migrate in through the cytoplasm, they tend to move centripetally. They move toward the nucleus. And as they do so, there are proton pumps in the membrane, among the membrane proteins, that cause these to become more and more acid on the interior. As they pass further and further into the cell, they will also fuse with what are called primary lysosomes. These are other vesicles, membrane-bound vesicles, that often contain either the precursors of proteol or enzymes, often proteolytic enzymes, or the actual active enzymes themselves. When I make a point about precursors, is because sometimes it takes more acidification of this combined secondary lysosome before those enzymes can actually act. When they do act, they may do a number of things. There may be a number of things that happen. In the case of nutrient molecules, what you often see is a release of the contents of the structure that's bound, and we'll talk about LDLs in a minute, either the contents of that structure, or if you have something like transferrin, acidification, which is carrying iron, acidification will loosen the iron up. It will cause it to become unbound from the transferrin transport molecule, and when it becomes soluble, it can be transported through the membrane into the cytoplasm in the cell. In the case of hormones or hormone receptor complexes, they may be recycled to the surface, as is shown over here, where you get these contents or these vesicles actually moving back to toward the surface of the cell and being re-released. You can get recycling, where some of these vesicles will actually fuse with the Golgi and ER and be recycled to the surface. Okay. Here's exocytosis or reverse endocytosis. So exocytosis is equal to reverse endocytosis. And in this instance, you may have an undamaged receptor back on the surface. The other things that can happen is that these can be further digested. You can end up with material going to what amounts to a storage or a garbage can within the cell. Those are often multivesicular bodies within the cytoplasm of the cell. But the interesting part here is the movement of these nutrient or hormone complexes into the cell, partial processing, and movement back toward the cell membrane, where you may reuse or partially reuse the contents of the material you started with. Okay, so that's receptor-mediated endocytosis. So there has to be a receptor here, and you have to involve this coated pit endosome lysosomal pathway in this instance. There are other ways for cells to get materials inside, but this is probably the best studied, most well-characterized path. Okay. All right, let's go over here. No, I want that one. I want this one. And I want to go back to this. All right. All right. Now, steroids. All steroids are derivatives of cholesterol. Okay. So cholesterol metabolism is important. There are two sources for cholesterol. One is diet, and one is endogenous or internal production. This slide deals in large part with nutritional provision of cholesterol. Cholesterol in the diet normally comes in mixed with other lipids during the process of digestion. So dietary fat, it's going to contain lipids. Any other guesses as what are or what other lipids besides cholesterol would be contained in your diet? Triglycerides, saturated fats, anything else? Unsaturated fats, anything else? Trans fats, anything else? There's going to be a whole mixture of them. There are people who have made their careers looking at what fats are in your diet. I don't find that particularly exciting, but they do. Okay, but you get dietary fat. The way it comes in through mastication and digestion in the stomach, it ends up being broken up into what are called micelles. Does anybody know what a micelle is?
Fat droplet.
A fat droplet. That's good.
Like,
Okay.
it's like the phospholipid bilayer, but it's not a
Okay.
bilayer, it's just one.
Recognize
one layer.
whatever you take in is usually suspended in an aqueous environment. So do fats like to be in an aqueous environment? No, they don't. And they tend to aggregate as clusters. And so if you've got something like a fatty acid or triglyceride, it often has one end that's more polar than the other. And the long chains, say if for a triglyceride, are going to extend in toward other fats. And here you may have cholesterol, you may have cholesterol esters. You'll learn more about these. But you're going to have all sorts of, and you may have just straight fatty acids, but they're going to form these kind of clusters. Micelles, you actually know something about micelles if you've ever washed with soap. Okay? If you've ever washed with soap. Okay. Do you know what soap is?
Molecule of the hydrophobic and hydrophilic
Okay.
properties.
A soap or a detergent is usually something that allows lipids to be suspended in water, and usually it has the shape where you've got one end that's charged or hydrophilic, and the other end is hydrophobic. And this is often a long chain, for example, the long chain fatty acid. So, and this is the other part of the molecule. So this associates with water. And this associates with other fats. Or oils or lipids. This is a detergent. Or a soap. Okay? So, a lot of the fats in our diet behave a lot like detergents. They also behave that way in aqueous solution in forming these micellar structures. When those micelles of fat get into the intestine can bring those across the cells, or it can bring them into circulation across or into the blood vessels that come close to the surface here. And as they do, they bring them in as large micelles called chylomicrons. And the chylomicrons that you see initially are coated with a few different proteins. So the chylomicrons differ from regular micelles in having a protein coat. So APO, what have I got there? ApoE, C2, B48. These are protein coat. So protein coated micelle equals chylomicron. As you go across this chart, what you're going to see are smaller and smaller lipoprotein complexes. These are lipoprotein complexes. They all have the general structure of protein-coated micelles. Fat in the middle with selected proteins on the surfaces. Now, if I take a blood sample, I'm going to get some of each one of these in circulation. If I then put these on a sucrose gradient or a density gradient, what happens if I spin a sample through a sucrose or a density gradient? Any guesses? If you, okay, the way a sucrose density or any other density gradient works, okay, you've got a test tube. And you put sugar or something else that will form a gradient. and it's less dense at the top and more dense down at the bottom. And the difference can be quite striking depending on what percentage of sucrose you use. So this would be a sucrose gradient. If I put my blood sample on top of this, which contains my mixture of chylomicron and other lipoprotein complexes, and then I spin this, at high speed, and when I say high speed, I mean 100,000 times gravity. What do you think is going to happen to the proteins in that mixture I started with? Are they all going to be in the same place? Okay. So what do you think is going to, how are they going to be distributed? Okay, so if I were looking for ribosomes or something which are really quite dense, they'd be at the bottom of the gradient. If I were looking for cell membranes, they'd be at the top of the gradient because there's a lot of lipid there. So if this is high protein, this is high lipid, what's the order of those lipoprotein complexes in the gradient? What's at the top?
Chylomicron.
Chylomicrons. Why do you say that?
Well, they're ultra low density. And
Okay.
They have lots of fats.
There are lots of fat. What you're seeing in this gradient, you're going from more fat to very little fat, and you're going across. You may maintain about the same amount of protein, but the ratio between the fat and the protein goes down. So if it's high fat, it's going to be up here. So chylomicrons are up here. Where are the HDLs going to be?
Way down
At the top?
there. Way down there.
HDL is going to be at the bottom. Next is LDL, IDL, VLDL, and remnants in here. But you've got this gradient going on. And as a matter of fact, at least for quite a few years, that's the way those fractions were generated before they did a cholesterol measurement on each one of them to tell you how much HDL cholesterol you had. Okay. So, what's happening here? Well, as these come into circulation, they encounter lipoprotein lipase, which can act. There are spaces between these proteins, so those enzymes can start to act on the contents of the micelles. And they will release free fatty acids, glycerol, to circulation. The remnants here with the certain proteins bind to receptors on the liver, and you get release of dietary cholesterol into the liver, other fats into the liver, and you get a reshuffling of the proteins on the surface as some of these are put back into circulation as VLDLs. Same kind of processing. Remember, it's the same vascular system. So you get more release of fatty acids, smaller complexes formed. Some of them get recycled again through the liver. Some of them get bound to those tissues that have the appropriate surface receptors. So if the tissue has an ApoE receptor, those complexes can bind and be taken in via receptor-mediated endocytosis to those tissues and supply those tissues with the appropriate complex of fats. Animals differ as to, for instance, which lipoprotein complexes they use most as sources for steroids in their steroidogenic tissues. Rats, for instance, are HDL animals. They bind to the proteins in the surfaces of HDL and harvest the cholesterol they need to make other steroids from those complexes. Humans are LDL animals, and they make use of LDL in circulation as a source for cholesterol for their steroid-producing tissues.
So LDL is the good cholesterol?
LDL, no, HDL is the one that's good cholesterol. It's only good because there's less cholesterol in it. And because we don't use this as a source of cholesterol. Cholesterol is not just a source of steroids. That's an important component of its functions. But it is also important in the formation of all cell membranes. Nerves don't work right if you don't have enough cholesterol. Now, this is cholesterol from diet. There are also enzymes in the liver primarily that will make cholesterol from acetate, a small 2-carbon monomer, effectively. So they're building cholesterol from acetate. You can get totally rid of cholesterol in your diet. Some people have tried that. You're still going to have cholesterol in circulation because you make it. Cholesterol can also be made in steroidogenic target tissues, but in much lesser quantities. So, just memorize this. This is de novo synthesis of cholesterol from acetate. It's actually from the activated form of acetate, which is acetyl-CoA. And what's happening is these pieces are being put together end to end. So you go from 2 to 4 to 6 to 8 elements. Then you phosphorylate part of it so you can couple it in a slightly different way. And you're building toward the formation of cholesterol down here. It's interesting in plants, in insects, and some other organisms. These intermediate stages are often routed to formation of other kinds of compounds, other kinds of secondary metabolites. Okay, that's biosynthesis. Yes?
Do we really have to memorize this?
In detail. No, you don't. What you need to remember is that you start with a small 2-carbon fragment to build this structure. This molecule is actually 27 carbons. So you've got at least essentially 14 of these these units being coupled together to form that. Now we get to the fun part. Who's had organic chemistry? Okay, you guys are going to help the rest of the people remember this. Okay. Steroid structures and steroid chemistry. All steroids are derivatives of cholesterol. They all have this 4-ring structure. These are fused rings. These are, in this molecule, they're unsaturated. That means they're saturated. All of the hydrogens, all of the bonds not shown are filled with hydrogen. This particular molecule, which I used to have people memorize, but don't worry too much about it, cyclopentanyl perhydrophenanthrene. The phenanthrene is this structure, ABC, and the cyclopentanyl is up here, and the perhydro means that all the bonds are saturated. Cholesterol is this molecule, 27 carbons. It's a sterol, which means it has an OH group on it. And the OH group here happens to be on this position 3. Now, I did not come up with the numbering scheme. Somebody else did. So don't blame me when it becomes not intuitive. But follow me and you'll see that it's not quite as bad as it looks. So if you start here at the top of this ring, this A ring, and you go 1, 2, 3, 4, 5. And rather than going there, you go 6, 7, 8, 9, 10. This is a naphthalene ring system. Okay. So 10 is there, 11 starts up here, 12, 13, 14, and then back 15, 16, 17. So there's 17 carbons in the central ring system. There are 2 methyl groups. There's one here at 18, and there's another here at 19. Then you're off on a side chain. So this is 20, 21, 22, etc., until you get to 27. Okay. Okay. All of the steroids are going to derive from beginning with this structure. What time we got? 0? Okay. I'm going to start with this next time, finish up, and we'll finish up this